This video focuses on electrophilic aromatic substitution on mono and polysubstituted benzenes, as well as two new nucleophilic substitution reactions, both involving aromatic rings. Up to now, we've only considered electrophilic aromatic substitution on benzene itself. When you perform electrophilic aromatic substitution on a mono-substituted benzene, you have the formation of a di-substituted benzene, and so you have to consider the formation of regioisomers, either ortho, meta, or para di-substituted benzene product. The substituent that is on the ring undergoing electrophilic aromatic substitution has two effects on the substitution reaction. It affects the rate of the reaction, either increasing the rate or slowing it down. And it also has a directive effect, having an influence as to which position on the ring the electrophile will substitute. We will examine both of these effects, and you'll see that the nature of the group attached to the mono-substituted benzene greatly influences the regioselectivity and also the rate of the reaction. The nature of the substituent on the mono-substituted benzene affects the rate of the reaction and the reaction regioselectivity. So while you do not get 100% of one isomer over the other two, there is some selectivity in the formation of the regioisomers. Here's what's observed. When you have attached to the benzene ring a strong electron withdrawing group, usually one that can withdraw electron density via a resonance effect from the ring. That is, that this substituent would be a polar functional group that has a pi bond conjugated to the ring. That will slow the rate of electrophilic aromatic substitution. And if you consider the general mechanism for electrophilic aromatic substitution, where the first step is the slow step, the rate determining step, and that step involves benzene acting as a nucleophile, it makes sense that if you have a group attached to the ring that's pulling electron density out of the ring toward itself, it's going to make benzene a poorer nucleophile and thereby slow down the rate of reaction. If the group attached to the ring is an electron donating group, for example an alkyl group, or even better, a group that has non-bonded electrons directly attached to the atom, directly attached to the ring, this will increase the rate of reaction. Again, that makes sense when you consider the mechanism for electrophilic aromatic substitution, that slow step, the first step, benzene acting as a nucleophile. If you have a group on the ring that is pushing electron density into the ring as benzene is giving its electrons to an electrophile, that would lower the activation energy. It makes benzene a better nucleophile, so that would increase the rate of reaction. However, consider the regioselectivity. When it's an electron withdrawing group attached to the ring, it directs meta. When it's an electron donating group that's attached to the ring, it directs primarily to the ortho and para positions. Well, there's no real intuitive explanation for that. Also, when it's an electron withdrawing group that's attached to the ring, that slows the rate of reaction, which makes sense. If that group has non-bonded electrons on the atom that's directly attached to the ring, that's also an ortho-para director. Again, doesn't make intuitive sense. So what we have to do is we have to come up with an explanation for this directive effect. One other thing I wanted to point out, a general trait, is that when you have a group attached to the ring that destabilizes a carbocation, that is, pulls electrons away from a carbocation, which would make it very unhappy since carbocations want electrons, they tend to direct meta. And when you have a group attached to the ring which can stabilize a carbocation by giving it electron density, they tend to direct ortho and para. Now, a question that sometimes comes up is when you have a mixture of ortho and para products, does one predominate over the other? And I took some information directly from the old chemical literature, which shows you that using alkyl groups as the substituent on a benzene ring, and those are carbocation stabilizers. You know that the more alkyl groups you have on a carbon, the lower in energy that carbocation is. Tertiary carbocation is more stable than secondary. So these are different alkyl groups, and they increase in size from ethyl, methyl, isopropyl to tertiary butyl. When the group is small, there's not too much selectivity between ortho and para, and so you get close to an equal mixture. However, when the group is getting bigger, there is a steric effect, and you can see we go from methyl to isopropyl to tertiary butyl, which you know is a big bulky group, and look, the amount of para increases in each of those two examples when the group gets bulkier. Before we proceed with an explanation of the directive effect of substituents on a benzene ring during electrophilic aromatic substitution, I wanted to show you some real examples, results from real reactions. The first two examples have a methyl and a methoxy group attached to the benzene ring. Both of these are electron donating groups. Both of them can stabilize a carbocation, particularly this one, the methoxy. It has non-bonded electrons on oxygen, so these could actually be used to directly stabilize a carbocation. In the case of toluene, methylbenzene, electrophilic aromatic bromination gives you very little of the meta, and you end up with, well, not quite equal amounts of the ortho and para products. Maybe there's a little bit of a steric effect. The electrophile is having a little bit more trouble approaching the ortho position because of the methyl compared to the para, but you end up primarily with ortho and para. And the same thing for, in this case, electrophilic aromatic nitration on methoxybenzene. We end up with a small amount of the meta nitro product, and we end up with primarily a mixture of ortho and para product. Now we go to a group which can withdraw electron density from the ring, and it's an electron withdrawing group. It's going to be a meta director, and it has no non-bonded electrons on the nitrogen, and so therefore we know it's going to give you the meta product. And you can see you get over 90% of the meta product and only small amounts of the ortho and para. One thing I also wanted to point out is that even though, as in this example here, you end up with a mixture of ortho and para products when you do this reaction, if you were going to use this reaction as a synthetic step in a multi-step synthesis, it's okay to say, I'm going to synthesize this paranitro anisole product from anisole. You would just have to indicate that this gives a mixture and that you separated this one away and you carried on in your synthesis with this one. So it's perfectly fine, even though you know you're going to get a mixture of products, to use this as a synthetic step, either if you wanted to make the ortho product or the para product. You would just indicate that both isomers formed and then that you just separated them in the lab and then carried on with in your synthesis with whichever isomer that you needed. How can we rationalize the observed directive effects of a substituent on a benzene ring during electrophilic aromatic substitution? We said that electron donating groups will direct primarily ortho and para, and electron withdrawing groups will direct primarily meta. There's two principal ways of doing that. One involves writing out reaction mechanisms for electrophilic aromatic substitution at the ortho, meta, and para positions on the monosubstituted benzene, and that's described in your book. Another way of doing it doesn't involve writing out reaction mechanisms at all. It's a kind of a quick way of doing it by drawing these charge-separated resonance structures, and that's shown on this slide. Remember, charge-separated resonance structures or charge-separated Lewis structures are poor Lewis structures. Over here, this ketone, this is the only resonance structure for this ketone. However, if I take the two electrons in the pi bond and I draw an extreme Lewis structure, a bad Lewis structure, and I actually push those two electrons toward the oxygen, extreme meaning that the oxygen is tugging at the electrons. It's more electronegative than carbon, so this is a polar pi bond. Well, give the electrons to oxygen and look at the Lewis structure. That Lewis structure says that this carbon has a positive charge and oxygen has a minus charge, which means that this carbon is electron poor. And you'll see in the chapters on carbonyl chemistry that this is a good way of predicting that nucleophiles will attack this carbon. And you'll see there's going to be a whole variety of nucleophiles that will be attacking that carbon. So please remember that these charge-separated Lewis structures are poor Lewis structures, and they're only just a trick, a technique, in order to gauge the chemical reactivity for various reactions. And we'll do that in this case here. All right, how can we use charge-separated resonance structures to explain that substituents like this, these strong electron withdrawing groups, those that they destabilize a carbocation, that they direct meta? Let's take a look at this example. This is an example of a strong electron withdrawing group. We have a polar conjugated system directly attached to benzene. The oxygen carbon double bond is conjugated, and therefore these electrons are being pulled toward oxygen. We can show that by drawing these resonance structures. The first resonance structure is similar to the one in the box over here, where I give the pi electrons in the carbonyl to oxygen. I end up with a positive charge on this benzylic carbon. If I have a positive charge on the benzylic carbon, I can now start to take electrons out of the ring and form a double bond to the benzylic carbon, which would give me a positive charge in the ring. If I continue drawing resonance structures in the usual fashion, you'll see that I end up with the positive charge on the ring in three specific places: ortho and para positions. So this is telling me that not only is this ring electron deficient, right? It's attached to an electron withdrawing group, making benzene electron poor, a poorer nucleophile. Not only is this ring electron deficient, but it's most electron deficient at the ortho and para positions. So if an electrophile was floating by, and an electrophile, as you know, is looking for electrons, where is it going to be most attracted? Ortho, meta, or para positions? Well, it should be attracted primarily to the meta positions, because even though the entire ring is electron poor, by drawing these charge-separated resonance structures, we've revealed that they're most poor at the ortho and para positions. So something that wants electrons is not going to go to those positions which have low electron density. It's going to go to the position which has a higher electron density, and that would be the meta positions. Let's turn it around and put an electron donating group, carbocation stabilizing group, attached to the ring. In this case here, it's a methoxy group. I'm going to draw charge-separated Lewis structures, charge-separated resonance structures, where the group that has electron density, the oxygen, it has non-bonded electrons. You might think oxygen is electronegative, but it's a carbocation stabilizing group. It has non-bonded electrons attached to the ring, which can be used in bonding. And so if I draw an extreme structure like this, and I make a oxygen-carbon double bond between the oxygen and the carbon on the benzene ring to which to which it is attached, I'd have to then break a pi bond in the benzene ring, and I'll localize this lone pair on this position over here. Again, I'll draw resonance structures in the normal fashion, and you'll see that not only do we know that this ring is electron rich because of this very generous oxygen donating its non-bonded electrons to the ring, we see that it's most electron rich at the ortho and para positions, as indicated by the non-bonded electrons, the little minus charge I put there to indicate those carbons have an abundance of electrons. So it makes sense that an electrophile would be most attracted to the carbon that has the carbons that have the highest electron density. In this case, it's the ortho and para carbons and not the meta. How about this example here? Chlorine, you know, is electronegative, and you would think that, okay, maybe it is pulling electron density from the ring, it slows the rate of reaction, but why is it also an ortho-para director? And the key thing here, the answer is the non-bonded electron. So it doesn't matter what the nature of this group is. If you think of something as electron donating or electron withdrawing, beyond its electronegativity, check to see if it has non-bonded electrons on the atom directly attached to the ring. And you'll see that, again, I'm going to draw these extreme resonance structures. Right? These are bad Lewis structures. However, they tell me that if an electrophile was going to come up to this ring and look for electron density, it's the ortho and para positions that are the most electron rich. So we didn't write out any mechanisms here. All I did is I just took the structures that we're examining, the ones that are undergoing electrophilic aromatic substitution, and I drew these charge-separated resonance structures, and I looked to see which atoms in the ring are most electron-rich in any of these cases, and those are the positions to which substitution would occur predominantly. So the summary here would be that ortho-para directors are going to be any substituent, either an electron withdrawing or electron donating substituent, that has non-bonded electrons on the atom directly attached to the ring. So even though you might think halogen is withdrawing electron density via an inductive effect, it can add electron density to the ring via this resonance effect, a bonding effect. Ortho-para directors are basically any substituent that stabilize a carbocation by induction or resonance. And meta-directors are simply just any electron withdrawing group without non-bonded electrons on the atom directly attached to the ring. For example, carbonyls, nitro groups. So if you're trying to remember, or if you're given a structure and you want to figure out whether the substituent on the benzene ring is going to direct ortho, para, or meta, check to see which category it falls into. A second way to explain the directive effect of a substituent on a benzene ring during electrophilic aromatic substitution is a more rigorous method, and it's the one that's described in your textbook. What is the method? Is first, for whatever monosubstituted benzene that you're analyzing, draw out the resonance stabilized carbocation intermediate leading to each regioisomer of product. So for aniline, you would perform ortho attack, draw out the carbocation intermediate. Para attack, again, draw out the carbocation intermediate. Meta attack, what's that carbocation intermediate? You then have to analyze these carbocation intermediates, compare them. The carbocation or carbocations that are most stable will be the easiest to form. That is, they will have the lowest activation energy for their formation, and it makes sense that the corresponding products will be favored. So what you should do for what's shown on this page is use this method to explain why the major product of electrophilic aromatic substitution on aniline are the ortho and para disubstituted benzenes. These are the major products. Uh, while the major product for electrophilic aromatic substitution with some generic electrophile on nitrobenzene gives you predominantly the meta product. And your textbook will provide an explanation within the chapter. What happens when you have more than one substituent on a benzene ring and you perform electrophilic aromatic substitution? Can you predict what the major product or products might be? Well, frankly, in some cases, you're just going to get a mixture and you really won't be able to predict. However, there are a couple of scenarios that will give predictable major products, and they're shown here. What if both substituents are orthopara directors, but they have different electron donating ability? Both the OH group and the ethyl group are carbocation stabilizers, they're electron donating groups, but only the oxygen, the OH group, has non-bonded electrons. The alkyl group does not have non-bonded electrons. That would make this group here the better carbocation stabilizer, the stronger electron donating group. Why is that? Well, how do we rationalize that an alkyl group stabilizes an adjacent carbocation? Remember, if you have a tertiary carbocation with 3 alkyl groups around it, it's more stable than a secondary which only has 2. So the alkyl groups are stabilizing the carbocation. You can explain it like this. You know, the carbocation is a p orbital. It's empty. It would love electron density. It looks next door, and we have a CH bond here. There are 2 electrons in this bond. They're tied up in bonding with the hydrogen, right? This carbon and hydrogen are bound together with these 2 electrons. If this orbital lines up a little bit and gives a little bit of its electron density, overlaps a little bit, that helps to satisfy this electron-poor carbon. But this can only be so generous. It cannot give its electrons to make a bond to the carbon. That would be breaking this bond between C and H. However, when you have an atom that has non-bonded electrons, they're non-bonded, they're just hanging around, they're donatable. So therefore, if you have a carbocation on the carbon next door to an oxygen or a nitrogen, atoms that have non-bonded electrons, or even a halogen, you can satisfy the hunger of electrons from this carbocation by actually making a bond between the oxygen or nitrogen or halogen and this carbon, and you end up with now the positive charge on the oxygen in this case. If that's so, the stronger electron donating group is going to have the biggest influence on the direction of electrophilic aromatic substitution. And you can actually write out the mechanism for electrophilic aromatic bromination on this ring at the ortho position here versus this ortho position that is ortho to the ethyl group. And you'll see that when you put the bromine here, you will end up with a carbocation that has 4 resonance structures. When you put the bromine here, or here, you'll end up with a carbocation that has only 3 resonance structures. So there's a real bonding effect going on here, which gives you a 4th resonance structure. More delocalization of charge. Remember the discussion of resonance structures. The more you can spread out a charge, the easier it is to form, the more stable that charged species is. So that's why this is doing the directing. A second example, a scenario, would be if one substituent is a meta-director and the other one is an orthopara-director. In this case here, the carboxylic acid carbonyl will have a meta-directing effect. It's withdrawing electron density via a resonance effect. And the bromine has non-bonded electrons. So if you had a positive charge on this carbon forming during electrophilic aromatic substitution, much in the same way that oxygen bonds to the positive charge. In this example, it could do the same thing with this bromine. Therefore, electrophilic aromatic nitration of this ring would be directed by the bromine. So the bromine is directing at the ortho to the bromine, these two ortho positions, and the para position. And so these are going to be the major isomers. Note that even though bromine is directing to this ortho position, you end up with a molecule which has three substituents all near each other. Due to steric effect, you have little to no nitration occurring at this ortho position. So in general, the stronger ortho-para director, the electron donating substituent, the stronger electron donating substituent has the greatest influence on the direction of substitution. Your book describes two types of nucleophilic aromatic substitution. This slide will describe nucleophilic aromatic substitution by an addition-elimination mechanism. Remember, in electrophilic aromatic substitution, you're substituting a hydrogen on the benzene ring by benzene acting as a nucleophile reacting with an electrophile. In nucleophilic aromatic substitution reactions, the ring itself is an electrophile. And in this case, in the addition-elimination type of nucleophilic aromatic substitution, you are substituting a leaving group on the benzene ring for the atom or atoms of the nucleophile. And in order for the benzene ring to act as an electrophile, normally you think benzene is electron rich, you have to have at least one electron withdrawing group present on the ring. And that electron withdrawing group, that strong electron withdrawing group, has to be either ortho or para to the leaving group. If it's meta to the leaving group, reaction will not take place. So the leaving group has to be para or ortho to the electron withdrawing group. This can be explained by the reaction mechanism. We have a specific electron withdrawing group here. This is a strong electron withdrawing group because it can withdraw electron density from the benzene ring via a resonance effect. In this mechanism, a nucleophile is going to attack the carbon bearing the leaving group. Now please, this is not an SN2 reaction. Please do not have the nucleophile attack this carbon and the leaving group leave simultaneously. You cannot perform SN2 or SN1 even mechanisms on an sp2 hybridized carbon. But what can happen here is you can make this new carbon nucleophile bond using these red electrons that come from the nucleophile. If you break this pi bond in the benzene ring, and I end up getting a minus charge, which I can delocalize around the ring. Now this reaction will only work if you have this electron withdrawing group directly attached either at the ortho or para position relative to the leaving group. And the reason is you end up with a fourth resonance structure. That means that the minus charge is further delocalized, which lowers the activation energy for the formation of this intermediate. So notice that I have a minus charge on this carbon, and if I make a new double bond between the carbon with the minus charge here and the carbonyl, I can break the carbon-oxygen pi bond and I end up with this. So this extra fourth resonance structure is only possible when the leaving group is ortho or para to the electron withdrawing group. And to convince yourself, you should take this substrate up here, where the leaving group is meta to the strong electron withdrawing group, attempt to do this nucleophilic aromatic substitution, and you will see that you will only end up with 3 resonance structures for the corresponding anion when the nucleophile attacks the ring. Once the nucleophilic attack occurs, and you end up with this resonance stabilized anion, the ring can re-aromatize by just taking a pair of electrons from the minus charge from this carbanion reforming a pi bond, and the leaving group will leave with its electrons. Overall, in 2 steps, right, it's not a one-step mechanism, in 2 steps, you've substituted a leaving group for the atom or atoms of a nucleophile. In this case, the benzene ring is acting as an electrophile and not a nucleophile. I want to emphasize once again the difference between electrophilic aromatic substitution and nucleophilic aromatic substitution. The main difference is in electrophilic aromatic substitution, the benzene ring is the nucleophile, that is, it's the electron donor reacting with an electrophile. In nucleophilic aromatic substitution, the ring is the electrophile, that's the electron acceptor reacting with a nucleophile. So a key limitation of nucleophilic aromatic substitution is that you have to have at least one strong electron withdrawing group attached to the ring, and that's why it's infrequently used in routine chemical synthesis. I want to show you, though, on the next page a field of research where it is sometimes used. Positron emission tomography is a functional imaging technique used to evaluate organ or tissue function. It's a valuable tool in medical research with applications ranging from research in Alzheimer's disease to drug abuse. The system detects gamma rays emitted indirectly by a positron emitting tracer on a bioactive molecule that's injected in the body. The tracer is frequently labeled with fluorine 18. 3D images of tracer concentration within the body are then constructed by computer analysis. The insertion of radioactive F18 into tracers is frequently done by nucleophilic aromatic substitution, and this is an example I took from the chemical literature. Here you see a benzene ring with a strong electron withdrawing group that has a leaving group in the para position. So the leaving group would be this amine. Nitrogen has a positive charge, and as it leaves, it would take the electrons in the CN bond. So this is a leaving group, much in the same way that when you protonate OH or OR, it becomes a leaving group. When this molecule was treated with F18, a nucleophilic aromatic substitution reaction takes place to make the F18 labeled aldehyde. They then reduced the carbonyl, aldehyde carbonyl, to an alcohol using chemistry that we'll see in chapter 18. Using chemistry you've seen before in Organic 1, you can do a substitution on this alcohol to make this alkyl halide. They treated this radio-labeled alkyl halide with this amine. It has a nucleophilic nitrogen. An SN2 reaction takes place, and they ended up making F18-FCP, which is a tracer used for PET imaging of dopamine D2 receptors. The second type of nucleophilic aromatic substitution described in your textbook is nucleophilic aromatic substitution by elimination addition. The example that you saw previous had an addition occurring before the elimination. In this case here, we do an elimination, and it's followed by an addition. You must have a benzene ring that has a leaving group with a hydrogen on the carbon next door. Treatment with strong base will do an E2 type of elimination, and you get a reactive species which is called benzene. The YNE at the end infers that this is a triple bond, and you can draw it as a triple bond, but the molecule itself, it has some diradical character. It's a weak pi bond here, very unstable reactive species, and it's definitely not isolable. So when it's generated in the presence of a nucleophile will attack one of the carbons of benzene. The electrons in this pi bond go to the other carbon. You get a minus charge, which picks up a proton from a proton source, and overall what you've done is you've substituted a leaving group for the atom or atoms that come from a nucleophile. You must keep in mind that nucleophilic aromatic substitution by elimination addition frequently affords a mixture of two regioisomeric products. As seen in this example, this aryl bromide, when treated with sodium amide, affords benzene, but benzene can be nucleophilically attacked at either carbon number 1 or carbon number 2 to afford these two regioisomers in almost equal amounts. Because of the fact that a mixture is afforded, this is usually not a common reaction in chemical synthesis, unless the number of possible products is restricted by the symmetry of the aryl halide or structural constraints. An example is shown on the next page. Alkaloids are nitrogen-containing compounds isolated from plants, and many of them are of biological or pharmacological interest. Nucleophilic addition to an arine was part of the reported total synthesis of the alkaloid cryptovoline, which was isolated from an Australian shrub. The researchers chose to synthesize this heterocyclic precursor to the alkaloid by nucleophilic aromatic substitution, elimination, addition because there was only one possible elimination, one mode of elimination that could take place with this aryl halide. Notice there's no hydrogen on the carbon over here, only over here. So this is the only possible elimination. This would be the only possible benzene intermediate that could be afforded, and this is the only possible mode of nucleophilic attack on benzene by this nitrogen because it's much closer to this, and there's no way it could reach around and attack this carbon. Once it attacks this carbon, you ended up with the corresponding anion, which gets protonated to afford the desired precursor.